Hello friends, first of all, welcome all into the first online tutorial video lecture of BSc Part 2 subject Microbiology. Today we are going to discuss about the syllabus or brief introduction of course number 5, that is a Microbial Physiology and Metabolism. It consists of 2 credits. First one, that is a Microbial Physiology, and second one, that is Microbial Metabolism. So in this present lecture we have to discuss about the unit number second or credit second, that is a microbial metabolism. So the microbial metabolism, it consists of 3 points. First one, that is a catabolism of glucose. Second one, fermentation, or B, fermentation, and C, bacterial electron transport chain, that is a ETC. So they let me introduce the first one, the point A, that is a catabolism of glucose. The catabolism or breakdown of glucose, it is carried out by the 4 different ways. First of all, that is a EMP, HMP, ED, and TCA cycle. So into that first lecture, we let's detail detail study of EMP path or glycolysis. So this glycolysis, that consists basically, in case of the all eukaryotic and prokaryotic cells, there are 2 types of pathways or 2 types of metabolic pathways. Initially, what is mean metabolism? Metabolism, it is a combined process of either catabolism or anabolism of specific biomolecule. So what is mean catabolism? Catabolism means breakdown of specific biomolecule, and anabolism means synthesis of specific biomolecule. So metabolism means it is a combined process of both of these, either synthesis or either breakdown. So into that, let me discuss about the catabolism and anabolism. So the metabolic pathways are of basically 2 types: the catabolic pathways and anabolic pathways. So catabolic pathways are involved in the oxidative breakdown of the larger complexes, and anabolic pathways are involved in the synthesis of compounds or synthesis of the biomolecules. For example, in case of the catabolic pathways, into this definition, it involved in the oxidative breakdown of the larger complexes. So the oxidative breakdown of the larger complexes means the specific proteins are converted into the amino acids. The larger polysaccharides are converted into the monosaccharides. Also the lipids are converted into the fatty acids. These are the catabolic reactions, or the specific linear chain of the any biomolecule. They will be converted into the monomers, or polymers get converted into the monomers. But exactly opposite to the catabolic, that is the anabolic path. So in case of the anabolic pathways, the specific types of the compounds are synthesized from this monomers. They get converted into the polymers or monomers. They will be, say, the different types of number of monomers, they will be linked in a specific chain, and they can form the polymer. It is known as a anabolic reaction, or this pathway is known as a anabolic pathway. Catabolic pathway is an exorganic in nature. Exorganic, this the specific types of the reaction, they have to release the energy in the form of energy currency, that is the ATP, NADH2, FADH2, like this. But in case of the endorganic reaction, the specific types of the pathways, they have to require the specific energy in the form of ATP, NADH2, FADH2, like this. So the endorganic reaction, the generally, the specific meaning of this exorganic or endorganic is that the exorganic reaction means they have to emit the energy or release the energy, and endorganic reaction, they have to require the energy. Exactly, the catabolic pathways are exactly opposite to the anabolic path. So, what are the characteristics of the metabolism? Metabolic pathways are mostly irreversible. These are they are carried out in only in a forward direction, no in a backward direction. Every metabolic pathway has a committed first step. The specific types of the, for example, glucose get converted into the glucose 6 phosphate. It is a committed steps. They have there is a no irreversible reaction. Glucose 6 phosphate can't convert into the glucose. So the glucose, they get converts into the glucose 6 phosphate. It is a committed step. All metabolic pathways are regulated. They are regulated by the specific types of the uh, interior cell intracellular messengers, and these intracellular messengers they will be regulate this pathway. Uh, for our well-known example is that in case of the human physiology, when we have to require the glucose, the glycogen get converts into the glucose. But if we have the body, they have the excess number of glucose, then this glucose get converted into the glycogen, and these are regulated by the two important hormones, that is the insulin and glucagon. So these are the regulated. The same this glycolysis or specific process of breakdown of glucose, they will be regulated by the different intracellular messengers. And the fourth one, that is the metabolic pathway in eukaryotic cells occur in a specific cellular locations. So each and every specific pathways they have the specific location. Either the some pathways are carried out in the nucleus, some pathways are carried out into the cytosol, or some pathways are carried out into the mitochondria. So the specific pathways they have the specific locations. Also they have the different types of the metabolic pathway in eukaryotic cell. They have the specific cellular locations. So let's we start the exact what is the process of glycolysis. So glycolysis means glycos. It is a Latin or Greek word. Glykos means sweet, and lysis means breakdown or splitting. The specific glucose there is the breakdown, or they will be converts or split into the two derivatives. These two smaller derivatives. From the glucose they get converts into the pyruvates, or these are the glucose. They have the six carbon compound, but pyruvate is a three carbon bond. The breakdown of glucose, or it is known as a glycolysis. So this pathway is known as a embedden Mayer of an Parnas path. It is a specific type of the pathway. It is also known as a EMP path. So glycolysis is also known as a EMP path, or embedden Mayer of an Parnas path. So introductory part of this glycolysis is that glycolysis is a sequence of the 10 enzyme catalyzed reaction that converts the glucose into the pyruvate with the simultaneous production of an ATP. It is a parallel or specific simultaneous reaction. They would from the it is the initial substrate of this reaction, that is a glucose, and the final product of this reaction, that is a pyruvate. So the glucose get converts into the pyruvate. So glycolysis is a sequence of 10 enzymes, and these 10 enzymes acts simultaneously, and they will be converts the glucose into the pyruvate, and why also they will be produce the ATPs. In oxidative process, one molecule of glucose is partially oxidized to two molecules of pyruvate. In case of this glucose, in this of this specific oxidative process, the one molecule of glucose will be oxidized to two molecule of pyruvate. The major pathway of glucose metabolism occurs in the cytosol of the cell. The glycolysis is a major pathway, or it is a universal pathway in each and every cell. The unique pathway occurs aerobically as well as anaerobically. It is a pathway that will be occurs aerobically, or in case of in presence of oxygen, also in case of the anaerobic means absence of oxygen. In both conditions, this specific pathway will be carried out and doesn't involve the molecular oxygen. In this specific pathway, no any involvement of the molecular oxygen. So it also includes the formation of lactate from the pyruvate. Also, these pyruvate they get converts into the lactate. The end pyruvate, that is the end product of this reaction. Also, this pyruvate they can convert into the lactate by the fermentative manner. The glycolytic sequence of the reaction differs from the species to species only in the mechanism of this its regulation and in the subsequent metabolic phase of the pyruvate formed. The glycolytic sequence reaction differ from the species. Each and every species they have the no any specific sequence, because different species have the different mechanisms, but the pathway is universal. But they have the difference in the specific mechanism or regulatory methods. So the species to species the pathway will be regulated differently. In aerobic organisms, the glycolysis is prelude to citric acid cycle and etc. The citric acid cycle and etc. they are the specific types of the chains. It is a continuous process. In case of the aerobic organisms, the glycolysis, the glucose get converts into the pyruvate. This pyruvate is cycled into the citric acid cycle. Then this citric acid cycle, the end product, that is a malate, and malate to the into this specific electron TCA cycle or citric acid cycle, they will be emits the NADH2, and these NADH2, these are chained, or they will be synthesized the ATP with the help of the ETC or electron transport chain. They will be situated into the inner membrane of the mitochondria, and they will be converts the NADH2 into the ATP. So glycolysis is the central pathway for glucose catabolism. Glycolysis is a central pathway for glucose catabolism. Centrally, they have the specific types of the pathway. They will be different types of the pathways are involved into the conversion of the glucose into the different products. But the specific glycolysis is a central pathway. So these glucose, they have the different types of the fates or different fates for the glucose. Glucose, they will be stored in the form of glycogen, starch, and sucrose. These glucose, in case of the plants, they will be stored in case of the starch or in the form of starch. Also, this same glucose, they will be stored into the glycogen in case of the animal, or this is also known as the animal starch. Also, this glucose is stored in the form of sucrose. So these are the storage form of the glucose. Then second one, the specific glucose, they will be synthesized the structural polymers. In case of the some gram positive as well as gram negative bacteria, they have the capsule or slime layer. This capsule and slime layer, both of these are made up of carbohydrates. These are the most of these heteropolysaccharides. Heteropolysaccharides, there are different types of the sugars are come together and form a specific layer, or this also known as a slime layer or capsule. The extracellular matrix and cell wall cell wall polysaccharides, there are most of these are the structural polysaccharides, or also the carbohydrates involved in the cell wall. These are structural polysaccharides. Most of the in case of the bacteria, the slime layer and capsule are made up of the heteropolysaccharides. So it is also one of the fate of glucose. Then also this glucose, they get converts into the ribose 5-phosphate. Ribose 5-phosphate. Glucose, it is a 6 carbon compound, and ribose 5-phosphate, it is a 5 carbon compound. It is a The specific glucose get converts into the ribose 5-phosphate with the help of pentose phosphate pathway or PP path by the oxidative manner. Also, this glucose, they get converts into the pyruvate. Glucose, it is a six carbon compound, and pyruvate is a 3 carbon compound. This breakdown of this glucose or glucose get converts breakdown of this glucose is known as a glycolysis, and this glucose get converts into the pyruvate via the process of glycolysis by the oxidative way, or by through the oxidation the specific aerobic way of glycolysis. The overall reaction of the glycolysis is that the specific glucose, they get converted into the glucose 6-phosphate with the help of the hexokinase. We have to initially discuss that the specific different types of the 10 enzymes are involved into the conversion of glucose into the pyruvate. So the first of all, that is a glucose get converted into the glucose 6-phosphate with the help of enzyme hexokinase. Hexokinase, it is an enzyme acts on the glucose and add the phosphate group at the position number 6 of glucose with the help of the cofactor, that is a Mn double plus. And they have to use the single ATP. This ATP get converts into the ADP. Then after all, the glucose 6-phosphate, they get converts into the fructose 6-phosphate, and the enzyme responsible for this reaction, that is phosphohexose isomerase. The isomerization of this specific substrate, that is a glucose, get converts into the fructose. Glucose, it is a aldehydic form, and fructose, it is a ketonic form. Also for this reaction, the phosphohexokinase isomerase, they have to require the Mg2 plus, it is an cofactor. Then this fructose 6-phosphate get converts into the fructose 1,6 bisphosphate or fructose 1,6 diphosphate. This fructose 6-phosphate, they get converts the ATP into the ADP, and the enzyme responsible for this reaction, that is a phosphofructokinase. This is a phosphofructokinase, and this phosphofructokinase, they will be converts the fructose 6-phosphate into the fructose 1,6 diphosphate. This fructose 1,6 diphosphate, they get converts into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Into this reaction, The aldolase, it is an enzyme, acts on the fructose 1,6 diphosphate, and they will be converts the fructose 1,6 diphosphate into the dihydroxyacetone phosphate and, and glyceraldehyde 3 phosphate. The dihydroxyacetone phosphate also they get converts into the glyceraldehyde 3 phosphate with the help of enzyme phosphotriose isomerase. With the help of enzyme phosphotriose isomerase. This glyceraldehyde 3 phosphate further converted into the 1,3-disphosphoglycerate. Converted into the 1,3-disphosphoglycerate, and they will be converts or they will be emits the one molecule of NADH with the help of enzyme triose phosphate dehydrogenase. Triose phosphate dehydrogenase. Also specified as the NADP, NAD they get converts into the NADH2. Then the 1,3-disphosphoglycerate they get converts into the 3-phosphoglycerate. 1,3-disphosphoglycerate they get converts into the 3-phosphoglycerate. At the position number first and position number third of this 3-phosphoglyceride, that is a both can carried out the carrying the phosphate group. This the first carbon phosphate they will be released out and they can synthesize the ATP with the help of enzyme phosphoglyceric acid kinase, phosphoglyceric acid kinase, and cofactor that is a Mg double plus. These 3-phosphoglycerate they get converts into the 2-phosphoglycerate. 3-phosphoglycerate get converts into the 2-phosphoglycerate, and the enzyme that is a phosphoglyceromutase. Enzyme that is a phosphoglyceromutase. This specific enzyme acts on the 3-phosphoglycerate, and this 3-phosphoglycerate get converts into the 2-phosphoglycerate. Then this 2-phosphoglycerate converts into the phosphoenol pyruvate with the help of enzyme enolase. And this enzyme they will be they have the cofactor Mg2 plus, and they will be released out the one molecule of H2O. That is a dehydration reaction. 2-phosphoglycerate get converts into the phosphoenol pyruvate with the help of enzyme enolase. They have to require the cofactor that is Mg double plus or Mg plus plus. Also they will be released out the one molecule of H2O. Then this phosphoenol pyruvate they get converts into the pyruvate, and the enzyme responsible for this reaction that is a pyruvate kinase. Also they have to require the cofactor that is a Mg double plus and K plus. In this specific reaction also they have to synthesize the second one ATP. The first one ATP is synthesized at the in this reaction, that is at the time of conversion of 1,3-diphosphoglycerate into the 3-phosphoglycerate. That is the first ATP. And in case of the second ATP, they will be synthesized in this at that step. The phosphoenol pyruvate get converts into the pyruvate. So from the one glyceraldehyde 3-phosphate, they will be synthesized the one NADH2 and 2 ATP. Also the dihydroxyacetone phosphate they will be converted into the glyceraldehyde 3-phosphate, and the same reaction will be carried out, and they will be synthesized the 1 NADH2 and 2 ATP. So totally they will be synthesized the 2 NADH2 and 4 ATP. But initially in this specific reaction, initially the cell they will be invested the 2 ATPs at the position of the first, that is a glucose. They will be converted into the glucose 6-phosphate. At that time they will have invested the 1 ATP, and the second ATP is invested when the fructose 6-phosphate get converts into the fructose 1,6 bisphosphate. So the initially they will be invested the 2 ATPs, and the gain is that the 4 ATP and 2 NADH2. So the net gain is that the for 2 NADH and 2 ATP. After when the one glucose molecule they will be converts into the pyruvate by the process of glycolysis. The specific cell they will be gain the 4 ATPs and 2 NADH2, but the specific cell they will be invested 2 ATPs initially. So because of this the specific cell they have the net gain of 2 NADH2 and 2 ATP. It is about the complete process of glycolysis. It is a brief introduction of glycolysis. Into the further lectures, we have to discuss about the in detail or stepwise the process of glycolysis. It is an only the overview of glycolysis. So, thank you very much.